Dear students, Assalamu alaikum and welcome to e-learning Al Noor International School Grade 12 Chemistry course. I'm your chemistry teacher Fraz Anjum. Today we will discuss Chapter 12, The Chemistry of Life, Lesson 5, Nucleic Acids, in the textbook page number 854 till 857. So please open your textbook, page number 854. The resources you need: textbook, notebook, pen, pencil, etc. The main objectives are of this lesson: identify the functions of DNA and RNA, identify the number of bases of DNA required to specify one amino acid in a peptide chain. And remember, this is 2-week lesson, so half lesson we will study in this week and half lesson inshallah we will study next week. Okay, let's start our topic: DNA and RNA. What are the functions of DNA and RNA? So the key function: DNA stores the information needed to make protein and governs the reproduction and growth of the cell and new organism. And the main function of RNA has a key role in the transmission of the information stored in DNA and in the synthesis of protein. So for RNA, protein synthesis; for DNA, storing the information and transferring from one generation to another generation. So from where we get the DNA and RNA? So more than 100 years ago, Swiss biochemists discovered a class of nitrogen-containing compound in the nuclei of the cell. These nitrogen-containing compounds called nucleic acid. So what are nucleic acid? Are the polymers that found primarily in the cell nucleus. So there are two kinds of nucleic acid in the cell. Number one, deoxyribonucleic acid, which is DNA commonly called, and ribonucleic acid, commonly called RNA. Let's start now the nucleotide. What are nucleotides? The monomers that make up DNA and RNA polymers are called nucleotides. The monomers, you know this one, monomer means single molecule, that make up the DNA and RNA polymer are called nucleotides. Such as this is called one monomer, other monomer, another monomer. So all monomer combine together and make the polymer, and this polymer could be DNA or RNA, like this. Okay, so nucleic acids are therefore polynucleotides. Poly means many, nucleotide means many nucleotides. So what are nucleic acids? DNA or RNA are therefore polynucleotides, means many nucleotides combine together and make the DNA or RNA, commonly called nucleic acids. Each nucleotide consists of a phosphate group, which is this phosphate group, a 5-carbon sugar, which is called pentose sugar. 5 carbon, 5, remember 5 means penta, okay? So pentose sugar, and a nitrogen containing unit called nitrogen bases, and this called the nitrogen containing unit called nitrogen bases. Okay, here adenine is there. Now when they combine together, means phosphate group combine or connect with the deoxyribonucleic acid, deoxyribose sugar or pentose sugar, connect with nitrogen bases, one of them, adenine, guanine, cytosine, thymine, among them, one of them. Okay, connect and make a group of monomer. This is the monomer. Okay, so this monomer called nucleotide. Okay, and then these nucleotide connect each other and make a DNA single strand. and then both strand combined together make the double helix strand. What is it? We will discuss. Just wait. Okay, let's start the explanation of nucleotide. So one of the component of the nucleotide, and most important, nitrogen bases. So there are 4 different nitrogen bases in DNA: adenine, guanine, thymine, and cytosine, and uracil in RNA instead of thymine. Means instead of thymine, uracil present in the RNA. Okay, so overall 5, but in the DNA 4. Okay, you can say like this, don't be confused, that in DNA 4, RNA as well 4, but thymine not present in the RNA and uracil not present in the DNA. Remember this. So these 4 bases abbreviated with A, G, T and C and U for the RNA respectively. So this is the structure of adenine and guanine, cytosine, uracil and thymine. You notice there that adenine and guanine both have double ring. One ring and other one. One ring and another one. So double ring carbon. Okay, double carbon ring present there in the adenine and guanine. And cytosine, uracil or thymine all contain one ring or single ring structure. Okay, let's explain the sugar. This sugar or pentose sugar present in the ribose form or in the deoxyribose form. What is the difference between them? So before we start this, first you have to know what is pentose. The sugar found in the nucleic acid are pentose sugar, which is a pentose sugar have 5 carbon atom. Means the sugar have 5 carbon atom. Let's count, check here, 1 carbon and 2, 3, 4 and 5. So 5 carbon. So that's why it's called pentose sugar. And remember in the class we discussed that aldoses and ketoses. Okay, so O's for the sugar. Okay, a combination of a base and a sugar is called nucleoside, not the nucleotide. Nucleotide means all 3 in the nucleotide, all 3 components present, like a phosphate, sugar, pentose sugar and nitrogen bases. Combination of this monomer called nucleotide. But if you remove the phosphate, so what left? Go ahead. Yes, one is nitrogen base, other one is pentose sugar. So combination of the pentose sugar and the nitrogen base called nucleoside. And this nucleoside normally we use in the preparation of the medicine in the pharmacy or pharmaceutical company for the antiviral or anti-cancer diseases. Ribose. Okay, ribose sugar found in the RNA, and this is the normal sugar with 1 oxygen atom attached to each carbon atom. Like a normal, the normal sugar have simple 1 oxygen always present there. In the deoxyribose found in the DNA, and this one is modified sugar. What is mean by modified? So lacking 1 oxygen atom, hence the name deoxy. Means D means less, without. Okay, so deoxy means is going to be remove 1 oxygen. Okay, so this is the difference. 1 oxygen atom is important for the enzyme that recognize DNA and RNA because it allows these 2 molecules to be easily distinguished inside the organism. Okay, so this is the identification of this. Now look here, both deoxyribose and ribose. In the second carbon, observe here, second carbon. In the second carbon, OH present. Okay, in the second carbon, OH present. Okay, this is normally is the pentose sugar or the sugar can always have oxygen. Doesn't matter in the form of OH, okay, or only O with like a connection. But here, look, this carbon present with attached with the oxygen. No, yes, this carbon attached with the hydrogen one side, but other side there is no oxygen. So this oxygen is going to remove, is not there present, not present here. That's why it's called deoxyribose. That's why it's called deoxyribose. This is the main difference between the deoxyribose nucleic acid and ribonucleic acid. Okay, so in ribonucleic acid, each carbon have one oxygen, but in deoxyribose nucleic, one carbon, which is second carbon, don't have one oxygen. This is the main difference. So I hope that you understood the pentose sugar and deoxyribose sugar and ribose sugar as well. Now, DNA and RNA structure. Chemists studying the nucleic acid discovered that the amount of adenine in DNA always equal to the amount of thymine. Means adenine always connected with the thymine. Means one strand is adenine, other side thymine. So remember with the word 8, like at what time, at 7 o'clock, 8, 5 o'clock, like this. So 88, remember like this, okay? And similarly, the amount of guanine always equal to amount of cytosine. Means guanine always equal to the cytosine. So G with the C, G with the C. And how you can remember that guanine connected to the cytosine? Government college, government G, college C. So government college GC and AT adenine thymine. This is the must be pair form. Okay, the combination how is going to be pair? Guanine always pair with the cytosine, not with the thymine. Okay, and adenine always pair with the thymine, not with the cytosine. Okay, so you have to understand the pairing or memorize or keep in mind that guanine with the cytosine, adenine with the thymine. So how you're going to memorize this? 88 and government college, like this. Okay, but in RNA, adenine equal to the uracil. Means there is no thymine. So instead of thymine, what going to be present here? Adenine with the uracil. And guanine with the cytosine, same as the DNA, RNA. In the RNA is there as well. So guanine, cytosine present in both DNA and RNA, but in DNA, adenine with thymine pair present. In RNA, adenine with uracil present. Okay, in DNA, adenine with thymine present, and RNA, adenine with uracil present. So remember this thing as well. Now this is the structure of DNA. Spiral. This is structure called spiral structure. Okay, this is structure. Okay, and this structure called double helix structure. Okay, so who proposed this? James Watson and Francis Crick. That's why it's called Watson and Crick model. Proposed that the structure of DNA consists of two polynucleotide, okay, polynucleotide chain wrapped into the spiral shape like this. So polynucleotide here, like a many nucleotide present here, and then wrap at the spiral shape like a rope. Okay, going to twist. Okay, this spiral shape famous like a double helix. This spiral shape famous in the like a double helix shape. So this is double helix shape or spiral shape. Alright boys, now in this DNA structure, every double ring base on one strand must be paired with the single ring base on the opposing strand. The pairing of A with T and G with C not only provide the best possible fit, it also allows the maximum number of hydrogen bond to form between the opposing bases. So look here. So this thymine going to attach with the adenine another way. Okay, this is the one strand. Okay, like this is the one strand. Okay, so adenine, okay, here adenine connect with the thymine, or thymine connect with the another side adenine, and one strand cytosine connect with the guanine. So this is called the pairing. So look here, one spiral another spiral. Okay, so these are nucleotide present here with a sugar and phosphate. Green shows the phosphate, okay, and pink shows the sugar, pentose sugar. So that's why it's called sugar phosphate backbone. Okay, after that this sugar going to connect with the guanine and then hydrogen bond, okay, and then going to connect with the single bond and another sugar from the other side. Okay, so like this they are going to connect each other. So here we go again, one more time. You can see here thymine, okay, so oxygen with nitrogen, between this hydrogen bonding here, the dotted hydrogen bond, and then from here another hydrogen bond and nitrogen prepared with the electronegativity difference more, so that's why it's going to be prepared the hydrogen bonding here. So two hydrogen bond means double hydrogen bond here, and here you can see how many hydrogen bond can be formed. 1 2 3 3 hydrogen bond can be formed. 1 2 and 3. So 3 hydrogen bond. Because of this hydrogen bonding, the molecule going to be more stable. Okay, that's why DNA structure is more stable. So remember the pairing, especially at and government college. Thank you so much, and we will continue this lesson in part 2. You can watch it here.